We're going to go ahead and get started, even while people are getting in, because there's a lot to talk about today, and we have a tasting involved as well. We have an hour and a half for this talk. I'm Anthony King. I have a small consulting company in the Valley and make wine for some small producers, and also work as the general manager at the Carlton Winemaker Studio, which is very fun to be fully involved with all those other great winemakers. Today we have two experts in their field, and we decided to do this seminar on the microbiology of wine really as a survey course, where I always feel like these seminars are about nuggets of information, and hopefully this will you'll get some nuggets of information that you either forgot or are new to you, but also just to complete the picture and help us all understand whenever something is going wrong in our wines. Our two speakers are James the Microbe Hunter Osborne from Oregon State University. He's the extension specialist, which means you can call him even at 1 a.m. That's part of his job. Rich DiCenzo, I'm sure we've all talked to at ETS. He's actually the head of the microbiology department. We refer to him as the Brett Whisperer. That's not true, but that's what he wants. And we fully support him, how Tom Danowski's talked about how we pay for it with taxes. We pay for everything many times over. So we're going to go ahead and get started with James Osborne.
Great. Thank you for that introduction, Anthony. Yeah, you can call me at 1 o'clock, but I'm probably not going to answer. But yes, as mentioned, I'm the extension enologist for Oregon, so if you don't know who I am, if you've got issues related to on the winemaking side of things, I can be a resource and involved in a lot of different things here. So thank you for attending this last session on Wednesday. I hope the rest of the symposium has been great, and we're going to wrap things up talking about microbes, and particularly talking about spoilage microbes and where they show up, what do they do, how might we design a QA program around understanding better what these microbes are up to, so that we can prevent some of these issues that we're going to get to taste some of these wines later and see how these microbes can cause some damage to your wine. So we're going to start off right at the start, and we're going to work our way through to bottling, and Rich and I are going to cover different aspects of this. What I'm going to mainly cover is kind of just a general overview of the main players. What are the main organisms that are there through these various stages of the winemaking process? When do they show up? Why do we care about them? And Rich is going to kind of fill in a lot of how we're sampling for them, what do some of these numbers mean, what should we be looking out for, and various things. So as a lot of just about everything related to winemaking, it does start in the vineyard when we're thinking about microbial load coming into the winery, what microbes are there, how many are there, and what actually make it through into the fermentation vessel. So intact grapes, certainly early in the ripening process, are actually, it's a very specialized kind of niche for microorganisms to grow and survive on. So there's usually fairly low populations if the grapes are nice and healthy and intact. But as we head to ripening and maybe over-ripening, or if there's any damage to the berries, be that through fungal infections or rain damage or birds or whatever can impact the integrity of that grape, then all of a sudden conditions can change very rapidly, and there's more nutrients available, and a whole host of microbes can grow and can cause some issues even before you get to the fermenter. If you're surveying what's on those grapes at any point as you get closer to harvest, it's going to change. There's a large amount of different fungal species and bacteria species that you can get from those grapes. I don't know how many of you attended the vineyard microbiome earlier today, but there was a lot of talk about the microbiome of the grape cluster and what's there. There can be a lot of different organisms there. Most of them don't survive through the fermentation process because of the low pH of the juice, the high osmotic pressure, and then later ethanol. So we start, well, there might be a large amount there when we're talking, particularly when we're thinking about spoilage organisms and organisms that are going to cause problems later. It kind of narrows down into some select groups of yeast and bacteria that we sample for, that we analyze for, and that we care about what they do. So One of the main groups of yeast that can be on the grapes close to harvest are going to fall into what we kind of call semi-aerobic yeast, so yeast that really, most of the metabolism involves oxygen, so they require air. These are yeast like Pichia species and Candida species, some Cryptococcus or Deboromyces. The Pichia and Candida species, some of those can carry through into the aging process, and so when we talk about film yeast, often there's a Pichia or Candida species, and some of those can come in from the grapes all the way through into the cellar environment. Now, once they're in that environment, that they may set up shop for a while there, but some of these originate in the vineyard. Then we have a group of yeast called the apiculate yeast, and these are probably the most common yeast and ones that, if you've ever done any analysis or sent out for analysis, yeast that you'll often find on the grapes. And these are particularly Hansenia spora uvarum, or sometimes known as Clochera apiculata. This is a yeast that's very common and often associated with VA or EA issues during cold soaking, during early fermentation. So very common. If you're going to find a yeast on your grapes early, close to harvest, it's more than likely going to be a Hansenia spora.
Is there a low temperature range for that shuts it down?
So they're from a temperature tolerance point of view, they're more temperature tolerant than most of your other non-Saccharomyces and Saccharomyces. So yeah, if you go cold enough with the cold soak, you'll slow them down. But if you're in the 8 to 10 Celsius, Fahrenheit, I'm still working Celsius, there'll still be growth and there'll still be considerable growth. You get above 10 and they can really, really take off. Getting down below 8 is not necessarily practical with cold soaks, but that would slow everything down. But when you're looking at the different yeasts that are there, these are the most cold tolerant that you're probably going to find, and somewhat SO2 tolerant compared to the other non-Sacs that are there. Now they do have a distinctive shape, and I hope you can see the shape of them here in the background. They kind of have a drawn-out lemon shape. That's why they're called apiculate yeast. They bud from each end rather than all the way around, and so they are a yeast that, if you have a microscope and you're doing some scans of juices, they are one of the few yeast you might find that you may actually be able to positively identify based on shape. A lot of the other yeast, if you look at the photos that I'm showing, they all kind of just look like ovoids or circles, very similar shapes, so it's very hard to distinguish what they might be. The apiculate yeast are ones that have this distinctive lemon shape, so if you see that, you can use that diagnosis from using a microscope. Most of the other ones, you're going to be guessing. So we moved on from the apiculate yeast to yeast that have some fermentative activity, might actually persist for a while. Some of these names you might recognize if you have been using any of these non-Saccharomyces cultures that are commercially available now. So Laccancia used to be known as Cloveromyces, Turulaspora, Menschnicoia. Those are cultures that you may be familiar with if you're using those. They have some fermentative activity, low fermentative activity, but they can persist for a while. They're not as reliant on having a lot of oxygen, a lot of air. And then there's Zygosaccharomyces. This particular yeast can be problematic post-fermentation or during aging if you have residual sugar, or it's an issue with people using concentrates, storing concentrates, because it's very resistant to high levels of sugar, so it can hang out in really high sugar situations. It's also fairly SO2 tolerant and ethanol tolerant, and can cause problems with re-fermentation if you have residual sugar later on. So that's one that can be problematic, and with paying attention to if you see these numbers and you know you've got some residual sugar. So that's the yeast side of things. On the bacteria side of things, when we think about the bacteria coming in on the grapes into the processing, the main bacteria that we care about or that we're concerned about are the Acetobacter and Gluconobacter. There may be lactobacillus species there, particularly if you've had damage to your grapes and everything's kind of started. You can get high lactobacillus numbers. There's some specific species early on that may cause problems. I think Rich is going to give an example later about a Kunkii, Lactobacillus kunkii, that can cause problems. So we're probably all familiar with Acetobacter and Gluconobacter. So these are bacteria that they're going to be there in low levels, low numbers, but again, any sort of damage to the fruit, or if things are hanging out at too warm a temperature in the winery before ferment starts, they can multiply and produce, obviously, acetic acid in the case of Acetobacter, but some other products as well.
I'd like to know more about that.
Which one? Tell me more.
Just lactobacillus, I was joking. Go.
If you have any questions at the end, you can ask them, Anthony. So that's kind of, if you take a snapshot of the major players, now there are others there, but the major players, the main ones of concern are your apiculate yeast and Acetobacter. Now, under certain conditions, the lactobacillus can also be problematic. But where we're most concerned with these organisms is early fermentation or pre-fermentation, particularly if you're doing any cold soaking or if you're holding grapes at a temperature that's warm enough for these guys to grow. If you're doing native fermentations, waiting for the ferments to take off with the resident Saccharomyces, if that is taking too long and you've got high populations of some of these yeast, then these are the yeast that are going to get going. And that's when you start to get that volatile kind of solventy notes coming off the top of the fermenter or the top of your cold soak. Hansenia spora uvarum is most likely responsible for those kind of ethyl acetate solventy notes. Acetobacter may play a role as well, though again, the oxygen kind of need for Acetobacter, if you're using dry ice or inert gas to blanket, you can reduce the risk of that. But Hansenia spora, if things are good for it to grow, it can grow rapidly and produce a lot of off odors and off aromas. The populations of these, of Hansenia spora and these other non-Saccharomyces tend to peak early, and then throughout fermentation they'll drop down. They may or may not persist post-fermentation, depending on the yeast strain. Acetobacter, very similar because it requires oxygen. Once you have this very anaerobic environment, you're going to see a decline, a drop in numbers, but that can pop back up later. And so your microbial load coming into the winery is going to impact things on the front end, but it can also impact things on the back end. So you have to be aware if you had problems early or you think you had high loads of some of these yeast or bacteria early, that they can still cause problems later. Just because you've got everything through ferment, there can be some carryover impact. Typical ways that we're dealing, that we can encourage their growth, which we're trying to do the opposite of low SO2 temperatures. If you're cold soaking, above 10 degrees, you're going to encourage growth. And then if you have any, for whatever reason, if you have a long kind of start to ferment, so if you're doing it native or even if you're doing inoculation and you do that incorrectly and you start with very low populations, that increase in lag phase there for the yeast to get up and fermenting presents kind of this window of opportunity for these yeast and bacteria. So limiting that window of opportunity there, particularly if you know it's a high-risk year or there's damage, you're working with damaged fruits, so you're probably going to have a higher population of some of these microbes. You wanting to limit that kind of potential for spoilage there, get ferment up and going, make sure you're keeping your SO2s, higher SO2 dosage, or keeping temperatures low for cold soak. The main spoilage compounds I've alluded to are ethyl acetate and acetic acid, and that's what is giving you the EAVA notes, solventy notes early in ferment. And that can carry through, if it's high enough concentrations, can carry through to the final wine. Some of it may blow off during fermentation. You may see a reduction in that, but if you have a lot of it on the front end, you can still have very high levels of these compounds, ethyl acetate and acetic, in the wine post-fermentation. The last point here to remember is that if you've got a lot of microbes in there and they're growing and metabolizing, they're also going to be consuming nutrients. And so the nutrients that you're trying to save for Saccharomyces to do the ferment, these guys are going to deplete some of those nutrients as well. So if you're doing an early, you've done a YAN analysis on the juice or the grapes pre-harvest or right at harvest, that can change rapidly if there's a lot of growth of these organisms during a cold soak, for example, because they consume YAN, they'll consume some of these nutrients. So timing of when you're measuring your YANs and your nutrient additions, keeping in mind that these spoilage organisms can also reduce and deplete some of the nutrients.
Do you have an idea of how much? Have you seen how much they can use?
Good question. I think Rich has got a slide later on about it. In some of our work, we've seen, a YANs decline by, 10, 15, 20%. It depends again how much growth you're getting and things like that. But they can, half the amount again if there's enough growth of them.
Easily less than that.
The bacteria alongside the yeast coming in on the grapes, the gluconobacter tend to predominate early. Once there's any ferment going, the acetobacter dominates later. They both can be involved in sour rot, so if you have damage on the grapes in the vineyard and you start getting basically a little mini ferment happening there, you get acetic acid bacteria that will be able to convert any of that ethanol into acetic acid. The gluconobacter also can produce other compounds, gluconic acid and other things that kind of contribute to that. So worst case scenario with these guys. And like I said, the high populations on the front end can have implications later on that you've got a high load of acetobacter, even though during alcoholic fermentation they're going to decline in population because of the anaerobic environment. They can cause issues later on. So key considerations pre-fermentation here. Keep in mind seasonal variability. So not every year is going to be the same. You're going to have different microbial loads coming in on your grapes depending on what's happening with the season. So warm, hot, dry seasons versus wet, damp harvests, very different grape conditions, very different microclimates. You're going to have very different microbial loads coming in. So what you saw one year is not necessarily what you're going to see the next year. And that goes for the chemistry of the fruit as well. And I know Rich is going to touch on that the fruit chemistries are going to impact how these organisms grow as well. And so those are going to change continually also. So seasonal variability can be huge. Processing conditions, how you're going about processing. Are you thinking about sanitation all the way through the process? Are you keeping your bins cleaned out when you've dumped them in? Are you cleaning them out so there's no juice sitting in the bottom of them starting to ferment? Are you keeping things at the temperature you'd like from a cold soak point of view or getting ferment up and going? Cold soaking, lots of considerations there. Should you be doing it given the risk factors? SO2, temperature, length, those are all things you need to consider based on the condition of the fruit and the microbial load that might be present. And then inoculation strategies, getting things up and going with inoculations, or if you're doing native fermentations uninoculated, maybe getting something going on the side, a PTQ, so that you could get things going if it looks like nothing's happening for a few days, being prepared to get that fermentation kicked off. And then never forgetting the interaction between the chemistry of the fruit and the growth of the microorganisms. And I know Rich is going to cover that in a little more detail about how those things are dovetailed together, and you can't ignore one or the other. You have to consider how those impact each other, particularly around pH and around SO2 use. Okay, so I'm going to hand over to Rich now, and he's going to talk a bit more about what happens in the juice and sampling at this point in the process.
All right, thank you, James. I'm noticing a trend, and that trend is this is the third time I've talked at this conference, and they always put microbiology, the last session, the last day. So I'm not feeling the love. I don't know what it is, but anyways, I'm really glad to be here, and I think James has just done an entire semester in 10 minutes. So what he asked me to do is drill down a little bit and provide some sort of real-world observations. So I like to start out with this slide, and it's pretty much what James said, is that juice chemistry impacts juice microbiology, juice chemistry impacts wine chemistry. Actually, everything impacts everything, all right? So we're not living in one little bubble. These things all interact with each other, which is why it's often very difficult to predict what's going to happen. And then it's always amazing that with all of this going on, you manage to make such beautiful wine. So we'll drill down into this a little bit. One of the most difficult things when we're looking at juice, and we start with juice, is how to get a consistent and representative sample from a vineyard. And it's critical if you want to have actionable data. And what does that mean? It means if you're going to pay for some analysis, you want to be able to use it. It has to be a tool that you can use, you can make decisions based on. How do you get, a representative sample from your vineyard? And that's not trivial, okay? So we have people send in clusters. We have people who send in a 60 ml tube of juice, which is supposed to represent 10 acres. Very, very difficult, okay? We know that, vineyards are not uniform, and whether it's soil moisture, soil composition, temperature, even microbiology. We have data to show that different parts of vineyards have different microbial populations. So you just try to do the best you can to get the representative sample out of the vineyard. And in doing so, if you're consistent in the way that you sample year to year, then that's a very powerful tool. It may not be the exact representation of what's out there, but if you're consistent, you can look at historical data and you can say, this year TAs were up a little bit, so maybe I don't have to add as much acid as I added last year. So a valuable tool, consistency. And why do we need to do this? Because guess what? You guys are not in the beer industry, right? You don't have the same raw material every year, right? You have grapes, okay? And grapes change, okay? So let's look at the juice. That's where we start with microbiology. As far as I'm concerned, we look at acid balance. It's very important, and primarily you might be looking more at what's my final TA going to be, but I'm looking at pH, okay? What's the pH going to be? We talk about sugar. We harvest, make some decisions based on sugar. I don't really look at it as sugar. I look at it as potential ethanol. The higher your potential ethanol, the more problems you're likely to have, the more difficulties, okay? And then nitrogen. And we call it yeast assimilable nitrogen, but it's also bacterial assimilable nitrogen. We could call it microbial assimilable nitrogen, but that acronym would be MAN, and I don't know, we might offend someone. So we'll just leave it at YAN for now. But when we look at this chemistry, and unfortunately, or maybe it's just the way that the system is set up, you spend a semester learning about chemistry, and then you spend a semester learning about microbiology. And we don't always put those together. So that's what I hope to do today. Okay, so with juice microbiology, when it's coming in, non-Saccharomyces yeast, as James just did a really good overview of that, they're critical. They're there. Lactic acid bacteria, they may not cause a problem early, but if they're there, they can cause a problem later. And the same thing with acetic acid bacteria. So when we have juice, it changes, okay? It is the source of nutrition for all these microbes, and it varies year to year. So to try to be a little bit Oregon-centric, I pulled up Pinot Noir juice chemistry panel data from the last 6 years. And so the pink is the low value for the 6 years, the green is the high value, and you can see it changes every year. Every year there's something different going on in the juice. And the changes can be significant. This year, we saw lower potassium values, lower pH values. And I'm going to show a slide on that right here. Small changes in potassium have a pretty good impact on pH. And so this year, what we saw, I think there was about 400 juice samples I looked at. The pH for 2018 on Pinot Noir was lower than last year, and certainly lower than 2016. And that's because the potassium values were lower. Now down in Napa, our Cabernet, which is usually pretty high pH, came in at about 0.2 of a point lower pH this year, and potassium was down about 25 to 30%. So once again, every year it's different. And every year I crunch these numbers and I give this post-harvest review, and there's always something different and interesting going on. So the reason we need to do this is simply because it changes. Now, when you look at microbes coming in on juice, this is just a random survey of some data I pulled up. The important thing is, I don't think I can hit it on this side, but on that last aisle, percent positive. So let's look at the percent of the samples that are positive, and this was not a large number of samples, 1,200 to 1,400, but 70% of the juice samples we looked at had acetic acid bacteria in them. We saw low numbers of Lactobacillus, Pediococcus, very low numbers of Brettanomyces coming in, but Zygosac, Pichia, and Hansennia spora are all there at very high numbers. So this is your raw material, okay? So you've got changing juice parameters that can, on a given year, support better growth, and then you've got all kinds of microbes coming in. Lactobacillus conchii. It's a strange one. We don't see it very often, but in 2014, we saw elevated levels of Lactobacillus conchii in California, Oregon, and Washington. Why is that? Okay, no idea. In many cases, malolactic fermentation was completed before primary fermentation began. I had a client that had a Sauvignon Blanc that went through ML while they were cold settling it, the juice. Okay, went through ML. So I'd never seen anything like that before. And when you look at the data, it was just striking that this is Oregon data in 2014, that Lactobacillus conchii was at much higher levels in the red. And we saw this across the board in California and Oregon. I have no idea why. Linda Bassan's group did a really interesting paper on Lactobacillus conchii, and they found that it's present in healthy beehives. It acts as a protectant. I think maybe it keeps fermentation from occurring in the beehive on the honey. But maybe we had a big bee year. Don't know. But anyways, that was something we saw. We did a study a few years back on non-inoculated fermentations in the Willamette Valley. And in 2014, we had 5 vineyard cluster samples that came in, and we did micro-fermentations. Only 3 of the 5 actually had Saccharomyces in them. So 2 of the vineyard samples, those clusters that came in, gallon bags, no Saccharomyces cerevisiae, okay? But we still identified 29 strains of native strains that year. In 2015, all 6 of the vineyard clusters that came in that year had Saccharomyces. The difference was I asked the winemakers that were participating in this trial to spread out a little bit and grab clusters from different sites in the vineyard, so not just a single location, one vine. And of course, we had more diversity. We had 32 strains that year. So out of 61 strains of Saccharomyces that we saw, we only saw 1 strain in both years. Okay? So that was out of the Highland vineyard, which had the most diversity of any vineyard I've ever seen. Pretty remarkable. But nonetheless, the microbial strains change from year to year, okay? So your juice chemistry is changing, your microbial chemistry is changing. And we also saw differences in non-Saccharomyces populations, sometimes pretty striking, so that was also fun. So why do you worry about vintage variability? Well, suppose you have a year where you've got data like this, and if you look at the bottom, the YAN number is pretty low, and then say you happen to have some Hansennia spora. Well, this was done on the 2nd day of cold soak. I can guarantee that by the time they inoculated, and this was a 4-day or 5-day cold soak, by the time they inoculated the Saccharomyces, there was no YAN. The question was, how much YAN can be utilized? Well, we often have clients ship us samples of juice from all around the country. And if you send samples to us, we use those Sarsted 50 mil centrifuge tubes, and those are pretty rugged tubes. Well, when that tube arrives and the whole top is bulged out, there's some pressure in that tube. And we've seen samples that come in with no YAN. No YAN, nothing. Okay, it's gone. And so we call the client and say, maybe if you need to resend that in order to get a reasonable number. So given the time, if a fermentation occurs, that population builds up. I like to say Hansennia spora is a nitrogen sponge, and it will use whatever it can. All right, thank you.
So we've got things successfully through alcoholic fermentation at this point. We've done all of the right things on the front end. We've minimized microbial threats, added enough YAN. Now we're moving into malolactic fermentation and then onward into aging. So what should we be concerned about then? What are the main players that show up, and how can we maybe prevent that or develop a strategy to prevent that? So post-alcoholic fermentation and aging, pre and during ML can be a prime time for certain organisms to grow in that wine. Now, depending if it's a red or a white, the temperature of that wine, if it's a red ferment, there's still quite a lot of warmth in those fermentations. There's still sufficient CO2 that it's still fairly anaerobic, it's still gassing off. So aerobic microorganisms early at this point are less of a concern because there's some blanketing there from the ferment. But there are organisms that can grow that don't need oxygen, or oxygen might be stimulatory but not really necessary. And this is that period of time, if you're going through malolactic fermentation, or you want to, where you can't be adding SO2 to protect the wine. Once you add SO2 into there, your task of getting through malolactic fermentation becomes infinitely harder. So you've got warm wine, end of ferment, nutrients around, no free SO2, and you're trying to get your wine through malolactics, so you don't necessarily want to chill it down too much either. So this is a period of time which can be a high-risk area. If malo doesn't get going, if the wine sits for a long time, warm temperatures, other things will get going. Post-malolactic fermentation, once you're through that process, then you can start the process of protecting your wine, getting SO2 on there. The CO2 blanket created during primary ferment and maybe a little during malo is going to have dissipated, so then you're going to have to start thinking about preventing these aerobic oxidative organisms from growing. The main player that we are concerned about, although I think we were talking with Rich about it before, as far as whether it shows up all the time or not, is Brettanomyces bruxellensis. So you've hopefully or probably heard about this particular yeast and the problems it can cause, the issues that can cause. The major issue with it is that it can grow on very little nutrients. It can persist for a long period of time. It loves living in barrels, which are difficult to clean and remove. And it can cause significant issues on the spoilage side. So from a nutrient requirement point of view, it needs very little sugar. So you may have a nice dry ferment and think you've got to a very low sugar level. I guarantee you there's almost always going to be enough sugar in there for Brettanomyces to grow well and cause problems. So starving it out from nutrients is not going to be a sole strategy. Now, if there are a lot of nutrients available, then things get even worse. But if your strategy is just to get the wine dry and it'll be protected, well, Brettanomyces will still be able to grow. And that goes for oxygen as well. It's not an aerobic organism. Oxygen or air can help stimulate its growth, but it will grow in your wine even if you have a very anaerobic environment and you do well with topping and things like that. So it's difficult when you have an infection of Brett to prevent it, particularly in a barrel environment where sanitation and cleaning of those barrels becomes problematic. So there's a nice
Brett, we all have more questions, but we'll wait till the end.
Yes, thank you.
Brett.
And more puns. I'm cluing on. Yeah. So Brett, the main thing we worry about with the Brettanomyces is production of a number of different spoilage compounds. And so the classic kind of marker compounds for Brettanomyces are the volatile phenols. So you have the 4-ethyl phenol, which has kind of a distinctive medicinal band-aid notes, and then you have 4-ethyl guaiacol, which is maybe more on the smoky, clovey note. And depending on the amount of the precursor compounds there, the hydroxycinnamic acids and other conditions, you can have a range of amounts of these compounds produced. There seems to be some variability with the Brettanomyces as far as maybe how much of some of these compounds are produced. So it's not always the same smell every time with Brettanomyces. It's going to depend on the wine itself, the matrix it's in, how it presents itself. There are also a lot of other compounds, such as isovaleric acids, some aromatic amino acids, and some other aroma compounds that I think we don't yet really understand well that also play a role in the sensory impacts of Brett. So we think mainly about 4-EP and 4-EG, but there's a host of other aroma compounds that can also accumulatively give you this Brett smell. Aside from Brettanomyces, when we're thinking about particularly pre- and post-malolactic, where maybe we're less concerned with the oxidative organisms, we have the lactic acid bacteria. So these again are semi-aerobic, anaerobic, so they don't require oxygen, microaphilic really. So a little bit is helpful, but they're going to be able to grow in your wine no matter what you do from a topping point of view. I didn't list Enococcus in there as a potential, but if you're not wanting your wine to go through malolactic fermentation, then Enococcus would be considered a spoilage organism. But we're mainly here, the other two lactics, Pediococcus and Lactobacillus, and there's a variety of different strains and species, particularly on the Lactobacillus side. There's a number of different species here that can grow. They can conduct malolactic fermentation even, so you may have a spontaneous malolactic that could be carried out by a Pediococcus or a Lactobacillus species that may lead to some off notes and aromas that you weren't planning with your malolactic fermentation. So if you have a spontaneous malolactic fermentation, you can't guarantee that that's Enococcus doing that. The spoilage compounds they produce are pretty wide and varied. Acetic acid is one if there's sugar around, if you have spontaneous malo sometimes, or like Rich was talking about, maybe malolactic fermentation occurring even before alcoholic, you often can see an increase in VA, volatile acidity. So that's coming from acetic acid, from sugar metabolism. Diacetyl, again, in some wines, low amounts, that might be a desirable thing, but high amounts of it in whites and reds can be detrimental. And with the diacetyl production here by Pediococcus in particular, we're talking about much higher amounts than the small amount you might get from your Enococcus strain that you're using to make the butter Richard. We're often talking much higher amounts that become pretty objectionable, even if you're after a buttery note in your wine. Then we have a group of compounds called biogenic amines that are derived from amino acids, certain amino acids. And so some of these bacteria, and it also includes some Enococcus, particularly if you're doing a native uninoculated, the strains of some of those are capable of producing these biogenic amines that can have some sensory impact, and then also concerns from just having very high elevated amounts of these compounds that are sometimes considered to have health implications. So biogenic amines are problematic, and then mousy type notes in the wine. So here it's, I don't know, have you ever had a nice mousy wine?
Lots.
Has a kind of a mouse cage taste, smell. I don't know about taste, but smells like a mouse cage in your mouth. And there's a number of different aroma compounds involved in that. It's not a common thing, but it can occur, and when it does occur, that's pretty detrimental. So,
And what do biogenic amines smell like?
They can have a range. The common ones, cadaverine and putrescine. So cadaverine and putrescine, those are kind of the dead body kind of.
Yeah, they're one of the things that we have observed, and we just had a wine last week that they had a differential response in the bottle. Some of the bottles were tasting have a metallic taste. And I associate that with high levels of putrescine. And so we looked at the two bottles, and there were the bottles with the aftertaste, the SO2 had completely disappeared. There was no SO2, so it might have been a cork seal or something occurring at bottling. And they had Pediococcus in the high 100,000 range. The bottles that didn't have that metallic taste had very low levels of Pediococcus and the SO2 they expected. So it can, at high levels, have a metallic taste.
Aftertaste, yeah. I've also seen it where it's kind of a masking of everything else. It's just a very muted, flat note. So some of these are overt, that's like really obvious, and others, it's just a masking of what was originally there. So you lose all of the lovely fruity notes in your wine, and it just has a very flatness to it. So it can be very subtle from that case through to very overt, like the mouse cage example. So those are Brettanomyces and lactic acid bacteria that really don't rely on having exposure to oxygen to be able to grow and multiply. And so your control there is around your SO2 management and pH management and sanitation. And then we have other organisms that are reliant on exposure to air. They're aerobic, they require oxygen. And so this is where, alongside the SO2 and pH management, it's also headspace management, preventing exposure of air during transfers and during transfers and filtering operations and anywhere where oxygen can get into the wine. So the main organisms we're concerned with are ones that can cause films on top of your wine, and they are either bacteria or yeast. And the film-producing yeast, again, Pichia or Candida species. Often the film there is more of a dry film with bubbles and stuff, but not always. And then you have Acetobacter, that again tends to be more of a thin, slimy film. Sometimes you'll see that around the wine-air interface. So if you've got a tank that's low, you'll start to see it around there, or they like to hang out around the bung of your barrels where there's lots of exposure to air, etc. So these guys can grow fairly rapidly. They can produce ethyl acetate and acetic acid and acetaldehyde, primarily from ethanol. And the other, apart from just the aromas that can be off-putting, they also will quickly, because of the production of acetaldehyde, quickly deplete your free SO2. So you'll see your free SO2 deplete. You do an add to try and get it back, and that will get used up and depleted. And you're kind of fighting the acetaldehyde game there, where it's binding that free SO2. And you're trying to control the microorganisms' growth with it, but they're producing this compound which is somewhat protecting them. So seeing large and quick drops in your free SO2, often indication of acetaldehyde being generated either microbially or chemically as well if there's air exposure. So key considerations at this stage in the winemaking process, like I mentioned, don't forget about what happened pre-fermentation, the load coming in on your grapes. Your chemistries there are going to influence what's happening to the wine during aging, and so shifting and changing management practices, thinking about those things. Problematic fermentations, be them alcoholic or malolactic fermentation, just provide windows of opportunity there for many of these microorganisms. So stuck fermentation sitting there for a long time, all of a sudden you start seeing the VA rise way up. You've probably got a bacterial contaminant there that's able to grow and taking advantage of the sugar that is around and producing VA. So these are wines that to take careful consideration of if you've had trouble getting them through primary ferment or through malolactic ferment. There's increased chances, increased risk of spoilage there. Sanitation, SO2, pH management, things that main strategies there, and then effective monitoring, knowing what you're dealing with from a microbial load and from the chemistry point of view, like what Rich was just talking about pre-ferment. There's key steps post-ferment that we're going to talk about in a minute, where monitoring them is going to be key in making some strategic kind of decisions on what to do with this wine, what treatments are available, and when should you be doing this monitoring. So we've gone through at this point, I think before we hand back over to Rich, we're going to actually taste through the wines that are in front of you. So there's kind of an order or a way that I'd suggest we do this. So what I've done is I've tried to create a few different microbially spoiled wines, and I've done that by, rather than spiking in the compounds, which sometimes is a bit hit or miss, actually inoculated a few different wines with some of these organisms, encouraged them to grow and do their thing, and then present them to you. So you'll find a range of things that you may or may not associate with some of what we've been talking about. The first 4 wines are a grouping. So the wine number 1 is a Pinot Noir that we're calling the Pinot Noir Control, and wine number 3 is a Cabernet Sauvignon Control. So what I want you to do is to smell, taste through the first 4 wines, with the first 2 being one pair, so compare and contrast the first 2, and then the second 2 compare and contrast those. So you've got a pair of Pinot Noirs and a pair of Cabernets. Take note of any aromas or tastes or aftertastes or anything lovely like that. Write those down, and in a couple of minutes I'll ask for some feedback about what you're finding in these wines. Feel free to point that out, because Britt can't do that. Okay, if we all had a chance at least to go through the first 4. So like I said, we inoculated, added in a particular organism to wines 2 and 4, and these are both 2018 wines, a Willamette Valley Pinot Noir, and it's a Walla Walla Cab that we had. It's kind of, the color in the Pinot this year was pretty crazy. Comparing the Cab and the Pinot, it's kind of look very similar. Let's start with wine number 2, and just if there's any descriptors that you guys were picking up, anything different compared to 1 and 2 that you thought was interesting or different about the aromatics or the taste in wine number 2. So anyone want to volunteer a descriptor or 2? A little chalky. Muted. What's that? Sweaty. Yeah, I think was that one you mentioned. You got a little.
Well, on the back end, mousy to me is an aftertaste because the compounds aren't volatile at wine pH, but when it's lingering in your mouth, it starts to become volatile. And to me, it's like at the back of your throat, somebody rubs a greasy Fritos corn chip. That's the best way to describe it. Okay, but I was getting it. What appeared there was some cheesiness, but maybe at the back of the throat, I thought maybe a little bit mousy. But
how about the Cab number 4? Any descriptors there that you noted or wrote down? What's that? Band-Aidy, a little Band-Aidy? Yeah. Johnson's baby powder. All right. What's that? Metallic. Yeah.
I got like a Jolly Rancher.
Jolly Rancher note compared to even the 3 and the 4. There was that difference.
On 4, there's definitely a synthetic sort of fruit that stands out. I don't know if it's watermelon or something in that range. I know that's not what we're looking for,
no,
but just a straight descriptor.
Volatile. Sorry, is there another one over here? Yeah. Old veggie. Yeah. Okay. So what we did with these wines, I don't know if we were successful or not, is we inoculated them with quite a high load of Brettanomyces. So I was joking that when you are now in on research, sometimes when we try and get these things to spoil wine, we have a lot of trouble. So maybe that's the key, is just give in. No. So yeah, we took Brettanomyces strain, we grew it up, we got it nice and ready to go in the wine, and we put it in the wine and left it in there for a few months. We were tasting it and noticing differences, and then we bottled it up and sent it here, and then I sent some wines off to Rich to get some numbers on it, and there was Brettanomyces in there, which was great, about 5,000 sows in both of them, but there was no volatile phenols, 0 for EP and 0 for EG, which was interesting. But I think there are some definite differences. I heard sweaty before for the Pinot, and I think, although not our purpose, that the Band-Aidy medicinal over the top can definitely be there, but even subtle differences with Brettanomyces growth, what we're seeing here, I think there's some of these other compounds that are impacting its growth. So this isn't what you would call a classic kind of Band-Aid medicinal note in either of those wines. And if you'd never had any exposure or experience with Brett smells before, it might just seem like just a little off, and you can't really put your finger on what is off about it. But there are some differences there. So,
was the VA affected at all?
A good question. I'm not sure I've got the VA numbers on.
No, we didn't run VA.
Yeah, but there were a couple of comments, I think, on so I might go back and check that. So those are the first set. I don't know if you've all gone on to the last 2, but if you haven't, taste and smell the last 2 wines, and then I'll come back and we'll talk a little bit about those 2. One of them is not as subtle as these ones, so hopefully you will get that one. Okay, so let's discuss the fifth wine. So this is a 2018 Pinot Noir. It's the same wine as the original control there. So if you wanted to go back and forth, that's another way to look for differences. Any aromas or tastes that stood out with that wine? Anyone wants to be brave and throw out some descriptors? Balsamic. Rotten cilantro juice, is that? That doesn't sound nice. Okay. Fake fruity candy. Yep. Butter. Yeah, I got initial butter hit off that. Anything with the taste or aftertaste? So this 2018 Pinot Noir, we loaded it up with mainly Pediococcus, although we threw some Lactobacillus in there just in case, to try and get some lactic taint. Now, lactics can present spoilage a full array of different things. It's really hard to pin down a specific lactic taint to a specific growth of a bacteria. In this case, one of the Pediococcus we used, and we got pretty good growth, like over 10,000 cells of PDO, and similar, I think, Lactobacillus. One of the PDO strains that we used, we know, is a very high diacetyl producer. So we got about 4, I think we got diacetyl measures, about 4 micrograms per liter. So it's probably right around the threshold with Pinot. If you're sensitive to diacetyl, you probably picked it up. In some previous work we've done with this particular strain, we got up to 15 micrograms per liter produced, which is well and truly above the sensory threshold. So one thing the Lactobacillus can do is produce excessive diacetyl, a lot more than you would necessarily get from Enococcus. I think you mentioned a milky kind of note to it as well. And so lactic funk it's a less well described phenomenon than breadiness, I think. So we often see a lot of range, and a lot can be attributed to, it's a little bit off, it's probably a lactic funk. So the more, exposure, something is off here, and without knowing what's growing in there, your chances of knowing what happened is pretty minimal. And then the last one, descriptors, smells, tastes, acetaldehyde. Yeah, this is the classic film yeast Acetobacter blend. This one we had no trouble getting going. We just left the carboy half-topped for a while, and voila. So I'm sure if you've ever experienced that, it's not difficult to get film yeast or Acetobacter going, but it can happen very rapidly. In this case, lots of acetaldehyde. It would consume up your free SO2 pretty quickly, and difficult to get back under control, and getting your free SO2 levels up to a level where it'll protect that wine now. So you got this double-edged sword of having the microbial growth and spoilage, and then also having to deal with acetaldehyde consuming all your free SO2. So I hope that was maybe some different spoilage than you've tasted or tried before. If you don't know what Band-Aid in a wine smells like for 4EP, 4EG, I'd be happy to supply some, or come to another one of my workshops and we'll jack up some of the Band-Aid. In this case, I think it was maybe some of the other notes that we highlighted.
If you're having trouble with microbial issues in your winery, have James make the wines, and they'll be clean.
Yeah, seems to work well.
So go
That's
ahead.
what I'm trying not to. And yeah. So I don't know about how long, because we just took one at the end point, and Rich is going to talk about representative sampling, and I think we did a poor job of sampling this when we sent it, but we had about 5,000 cells per mil in this, so it's kind of a mid. I think that's the question, like how much, is 100 cells a problem, is 5,000, and I think those are questions that Rich might address or be able to address. So it's probably right in that zone. Maybe we leave this for another couple of months and that 4EP, 4EG starts to come out. We don't know at this point. So 2 months in there, we didn't get any generation, but I think we affected the wine in other ways. All right.
And we still have a fair bit more material. Make sure you're sending questions. If you have them, we'll also do live questions. And the question, is the Irish fellow single? That's not the type of question we're looking for.
Irish fellow. I'm mildly offended by that. Yeah. You could have at least said Australian. That's close-ish.
Irish. Okay. Yeah. Okay. All right. So what I'd like to do is now talk a little bit, maybe drill down into some of the issues we see, but I'm going to step back into some spoilage that occurs during fermentation, some of the problems we see during both primary fermentation and then at the end of primary fermentation, the end of malolactic fermentation. And then I was asked to talk about barrel aging, what problems occur, and then what do we look for at bottling. So a bit of material to run through, so we'll try to get this done so we have plenty of time for questions. So when you're trying to manage microbes during fermentation, once again, it's a combination looking at chemistry and microbiology. And under the chemistry, the acid balance, pH, is really important to know, because if you're trying to see what's going on, if pH starts to go up or go down, that can be dependent on potassium bitartrate dropping out and where your pH is starting. Malic acid, to me, is critical during primary fermentation. And why is that? Why? Because it's the canary in the coal mine, okay? If you baseline your malic and you know what it is, then if you start to have a sluggish fermentation, you can start watching it. And I'll demonstrate a little bit later why that's so important. But malic acid, something we don't usually think of when we're talking about fermentation. But once again, to me, it is the proverbial canary in a coal mine. Potential ethanol is important. If you're going to ferment up to 16, you better be ready for potential problems. You have to be very vigilant. Now, here I think where you're in the 14 and a half range, not as much as far as the number of problems that can occur, but certainly as you go to higher alcohol, you can have problems. And nitrogen, we won't talk too much about that, except that, as you know, Saccharomyces sucks up all of the ammonia, and what's left is generally the amino acids. And so at the end of fermentation, what you usually have left is amino acids, and bacteria and amino acids are always a problem, okay? So mousy, biogenic amines, those are all from amino acids and bacteria interaction. So yes?
Just to clarify, that the alcohol isn't the issue as much as just stuck fermentations and issues.
Yeah, because if you're looking, once again, you're trying to assess risk. So as your fermentation starts, if you know, and I have clients who routinely try to ferment up to 16%, or some even go higher. If you're going to have a high alcohol endpoint, then it's going to be difficult for the Saccharomyces to get there. It's going to take longer to get there. And if it's taking longer to get there, you have to be very aware of what else is occurring in the wine. Okay. So we're looking at non-Saccharomyces yeast as far as the microbiology, what's starting things off. If you do non-inoculated fermentations, then obviously that's what's driving the first half of your fermentation. If you're doing non-inoculated fermentations, it's good to know if you have Saccharomyces in there. Usually, I think the study that we did indicated that the house strains of yeast have a bigger impact on your fermentation than what's coming in from the vineyard, at least once it gets going. And so you generally will have some Saccharomyces in the house. I always worry about heterofermentive lactobacillus, especially if you start getting a sluggish fermentation or it sticks. And then Enococcus. It's not just something you add from ML. It can be in your house. As a matter of fact, most wineries have resident strains. They could be commercial strains that were used at one time. But if you have a stuck or sluggish fermentation, I look at Enococcus as a spoilage organism. Okay, right there I lump it together with the other heterofermentive lactic acid bacteria. So what are the microbial faults we look at? I think the number one problem when you look at primary and then at the end of malolactic fermentation is volatile acidity, so VA. And I say that with some confidence because in Northern California, we have 4 companies that make a very good living for the owners and all of their employees doing VA removal. Okay, so that means there's a lot of VA problems, but RO can solve those. Sulfides are a problem, maybe something that is beyond this discussion today, but they do occur. Ethyl acetate, as James said early on, Hanseníaspora, notorious for making ethyl acetate. If you try, chilling things down, it makes more if it's colder, so that can be a problem. Ethyl phenols, if you insist on using wooden fermenters, okay? We recently had a concrete egg that came out of the end of fermentation with about 50,000 cells per mil of Brett and just starting to get a little bit of 4EP. So if you're using something with a porous surface for fermentation, you always have the potential for Brett and ethyl phenols. And finally, biogenic amines, we generally see those in non-inoculated malolactic fermentations. And as James said, you can have a non-inoculated ML occur from Pediococcus. I've I've seen that in a Chardonnay, and I asked the winemaker, he said, well, it was kind of a slow starter. I said, well, how long did ML take? And he said, 4 months. Okay, so that's a slow start. But that was driven by Pediococcus, and yes, there were biogenic amines in that one. So I just wanted to showcase volatile acidity. That's my pet peeve of faults, okay? It is the most common, and in my opinion, the most preventable. But we're going to look in that gray area. Well, actually, I'll go through the process of winemaking. And so you can get VA produced early, pre-fermentation, from yeast and acetic acid bacteria. The highest level I've seen on incoming juice is about 0.7 grams per liter. But where we start to see it occur and where we get problems is often at about mid-ferment, when those non-Saccharomyces yeast, and in my experience, particularly Pichia, starts to get stressed out as you're getting into that transition where the alcohol is getting stressful, and they'll make VA and it'll jump rapidly up to almost a gram per liter. The interesting thing is it's self-limiting. It doesn't go higher. It just all of a sudden appears and then it stops, probably due to alcohol stress. The area we see the biggest bump in VA is at the end of malolactic fermentation, and that's naturally occurring. Enococcus can make about 0.2 of a gram of VA from a gram of sugar. So if you press and you say, my wine's dry, I'm going to inoculate, go through ml, and you have a gram of sugar, you can expect a bump of maybe up to 0.2 grams of VA. If you have a stuck fermentation and you stick it off to the side and say, I'll deal with that in December, the highest level of VA I've seen, and this is the truth, 18 grams per liter of VA from a wine that went through a spontaneous ML, and it was all Enococcus. There was no lactobacillus in the wine. And that's a third of the way to vinegar. Okay, so that's a pretty tough bacteria. So once again, Enococcus, our friend when it does what we want. Otherwise, maybe not so much. And then, of course, you can pick up VA from barrel aging, and I'm going to talk a little bit about that. Bottom line is VA is probably the #1 fault, and if you are vigilant and you're looking at chemistry and microbiology and aware of what's going on, it can be prevented. Okay, so I'm going to hammer on this because every time I have the opportunity to talk to a crowd, I bring this up. Because even though I've been preaching this for about 10 years now, in the Napa Valley, I still see people that seem to get surprised by this. So this is a case study that we did several years ago, many years ago actually. And this was a wine that was starting to slow down, and the client ran juice panel and a scorpion panel. And not much to look at. There was some Enococcus. One of the things that's interesting is we have clients that are really diligent about running analysis, but I think in the heat of harvest, they often don't look at the numbers very well. Okay, so this is a case of that. So 6 days later, they went ahead and they did another set of analyses, and the glucose fructose was dropping. Okay, now as winemakers, you guys are the eternal optimists. If sugar's dropping, that's all you're concerned about. Okay, it's good, nothing to worry about here. And they continued on, but it kept moving slowly. And at day 27, they started picking up some other notes, some acetic acid notes, but sugar was still dropping, and they were pretty happy. And they said, well, run VA, and they ran VA, and the VA was up to 1.5 grams per liter. Happened very quickly. Okay, and so my first question to them was, did it go through ML? And their response was, no, I didn't inoculate for malolactic yet. And I said, that wasn't the question I asked. I asked you, has it gone through ML? And sure enough, it had. And if you look down at the bottom, the Enococcus population blossomed, and so this happens too often. It is preventable. Okay, and we can talk about that later. But this is something, if you're a winemaker, I mentioned to you earlier that malic acid is the canary in the coal mine. Okay, you know your fermentations, you know fermentation rate, what you expect. If things start to seem to go a little slow, run a malic. Either send it in, do it yourself enzymatically, heck, do paper chromatography. Okay, but sort of baseline that. And if you see it starting to go down, then you need to know what microbes in there and what's going on. You can prevent this from happening. Okay, so we're going to jump forward. I'm only really talking about VA for fermentation, because most of the time that's the big one that we see. But during aging of wine, what What can't go wrong? I mean, volatile acidity, ethyl phenols, ethyl acetate, biogenic amines, all these things can occur, and generally you can get these non-Saccharomyces yeast, for example. You certainly can have Brettanomyces, that's a problem. Heterofermentive lactic acid bacteria, Saccharomyces, perhaps it's just getting stressed, Enococcus, all these things can cause problems during aging. The question I get most often is, what numbers represent a risk? That's what everyone asks. Well, is this number a bad number? It's a tough question because, at some points in the winemaking process, a low number of microbes or even a moderate number of microbes is not an issue. If you're going to bottle and you have 30,000 cells per mil of Acetobacter, but it's going to be bottled with a screw cap and good SO2 and consumed in 2 years, then I don't worry about that Acetobacter. But if you're going to barrel and you're just starting to barrel age a wine that's going to be in barrel for 18 months and you've got 30,000 cells of Acetobacter, you're going to fight that the whole way. Okay, so it really depends. Same thing with Brett. If you're 6 months in the barrel and you've got 40 cells per mil of Brett, who cares? Okay, you know it's there, keep an eye on it. But if you're going to bottle and you have 40 cells per mil of Brett, that's a different story. So the numbers that represent a risk, I generally tell people that once you get to around 50,000 to 100,000 cells per mil is when you can start seeing an impact, a sensory impact on the wine. We've done a large number of trials with Brettanomyces, both in wineries and barrels, as well as on the lab bench for clients. Generally, it's around 50,000 cells per mil where we start to see 4-EP accumulate. Now, there's a lot of variety, but I can tell you we've seen this over and over again. So that's sort of my magic number. If you hit 50,000 cells, that's time to intervene. Okay, intervention required, not ponder the problem. All right? So that's as far as numbers, what I think. So when you're going to, have wine aging for a while, or you're going to, bottle the wine, I like to think of things when we look at risk. That's what I help people do, assess risk. And you need to know the wine chemistry parameters and determine if that creates a risk based on the spoilage microbes present. So growth promoters, I look at this. What promotes growth? So sugar, malic acid, nitrogen, okay, oxygen. Certainly in a barrel, oxygen's a bad thing. Biomass. What do I mean by that? I mean other microbes. If you bottle a wine and it still has 3 million Enococcus in there, guess what? They're eventually going to die and release their cell contents. And microbes are cannibals, if you will. They eat what's there. So I look at high levels of other, non-spoilage microbes as a nutrient source, so that biomass. So those things are all growth promoters. Obviously, growth inhibitors, SO2, molecular SO2, high ethanol, low pH, lack of nutrients. Somebody once asked me if I was going to bottle a wine and I didn't want to use SO2, what kind of wine would I make? And I said, well, it'd be 17% alcohol with a 3 pH. And that'd probably take care of things. But in general, might taste like crap, but we'd be microbially stable. So bottom line is, when you're trying to make these decisions, you have to look at what microbes are there and look at, do we have something that will promote growth or something that inhibit growth? I have to make this brief public announcement, okay, about representative sampling of barrels and tanks. If you're going to spend the money for any kind of analysis, you need to know that you have a representative sample. Now, compounds associated with faults, things like acetic acid, ethyl phenols, ethyl acetate, sulfides, those are homogeneous, okay? They're well mixed. You can't help it. They're throughout the barrel or the tank. On the other hand, microbes are not. They stratify, okay? There's a thing called gravity, and it has an impact. And Brownian motion will keep some bugs up, but yeast, especially Brettanomyces, tends to settle down. I'll show you a very interesting slide about that. Acetic acid bacteria don't necessarily swim to the top because they like the top, but there's often oxygen there, and that's what helps them proliferate and thrive. So just really important. Barrels and tanks are individuals. If you're doing trending analysis, and I'm going to talk about that briefly at the end here, if you're trending, you can't trend using one barrel and another barrel and another barrel. If you're going to trend, you use the same barrel every time, because barrels are individuals, they're different, and you know that when you taste. Tanks, once again, same thing. So make sure you're sampling in a representative manner and you're consistent in where you're sampling. It is the most important thing for microbiology analysis. okay? And if you don't do that, then you're wasting your money. That's just my opinion. Okay, so I want to show you a little example of a sampling error that happened just with a high VA. Client thought he had a vinegar smell and he wanted to check for acetic acid and bacteria. First sample, his nose was right. The VA was up to almost 1.1 grams per liter. And so, but the Acetobacter number, he was impressed. He thought, great, I've got Acetobacter. But I looked at it and said, that's not right. That's a pretty darn low number. So he hadn't mixed the barrel, and he just put a thief in about halfway, sampled from the mid-barrel. So I said, just for kicks, I won't charge you for this, but why don't you just pull some off the top of the barrel, just the surface of the barrel, and rerun it. And it came back at 2.3 million acetic acid bacteria. Okay, they do stratify. And we talk about oily films. Well, there are oily films, okay? And there are also things that are worse than oily films. This here is a sample that was, the client said, I've got these pink chunks floating on top of the barrel, on the surface. I said, pink chunks? I said, could probably a floor yeast or something. I said, chunks? He sent it in. Those pink chunks were just masses of Acetobacter. Now, there was about a 6-inch headspace in the barrel, so it was abused. But anyways, the point is, if you do that, this is what you can expect to see, high levels. When we talk about acetic acid production in barrels and tanks, and if I hop on this acetic acid, it's because, once again, it is the most common and yet the most preventable. When you're in barrels, you get wine loss due to evaporation, okay? And, you can get, when I looked in the literature, 2 to 5%. And topping intervals vary. If you ask people how often they top, some people are right on a schedule, others kind of when they get to it, especially during harvest. But it's usually maybe 5 to 7 week intervals, maybe a little bit more. But oxygen ingress into the wine is a function of headspace volume, okay, time, and molecular SO2 levels in the wine. And this is really critical. Acetobacter needs about 3.5 to 4 milligrams per liter of dissolved oxygen before it starts converting ethanol to acetic acid. So 3.5 to 4. Most wines are, definitely below 1, generally 0.5, something like that in the mid-barrel. But let's talk about the surface of the barrel. Let's talk about the top 5 millimeters of wine. And 5 millimeters is what, 2/10 of an inch. So it's not very deep. Well, if you've got limited headspace volume and you have that short topping interval, and so you're putting less wine in, and you've got high molecular SO2 levels, then that air that's in that little surface area doesn't penetrate very far into the wine. But if you've got greater headspace volume, longer topping intervals, lower molecular SO2, the oxygen or the air can penetrate a little bit further. We're not talking where it's going to change the wine, but enough so you get proliferation of acetic acid bacteria on the surface. I have clients who say if they have to put more than 150 mils in, then they're waiting too long between topping. They record how much volume they put in every barrel. They're pretty fanatical. But then again, they don't ever have any problems with acetic acid bacteria. So just something for thought, okay? Going on to once again about sampling error. This was a really cool study that we did looking at long-term impact of various sanitation methods on barrels. And there was a winemaker who really didn't like to stir the barrels. She was the assistant, and every 2 weeks she had to stir barrels. And she had 32 barrels. Oops, I went backwards. And so if you look here, the red lines are the barrels after they've been stirred, and the green lines are the barrels before they were stirred, and this is levels of Brettanomyces. So in some places you have a hard time seeing the green line. If you don't stir a barrel and you're looking for Brettanomyces, you're unlikely to detect any more than, at the best, 5%. And most of the time it's well under 1%. Okay, so hard to make decisions if you're not getting a representative sample. Just something to show you. And in a real-world example of this, we had a client that purchased a bulk wine that had been tested for Brett and reported at 10 cells per mil. So they bought the wine and they bottled the wine. Post-bottling, it came back at 1,800 cells per mil. It's already in the bottle. Kind of hard to do anything. It's labeled, cork and capsule are on it. And so they said, well, how could that be? I said, I don't know. Let's talk to the people that you got the wine from and what they did. And they just took a sample from the top of the tank, okay? And that was that. So if you don't not mindful about stratification, you can have problems. That tank hadn't been mixed. The Brett obviously had settled down below the level of where it was detectable with our technology at 10 cells per mil. So that was a problem. Okay, so it does happen. All right, we'll wrap it up with recognizing risk at bottling. If you're driving down this road, and unless you were, one of those storm chasers you see on the Weather Channel, you would say there's some risk, and I'm going to turn around and head back the other way. At bottling, what do we look at for risk? What do I determine as risk? Well, high pH. What's that? Well, I'd say 3.7. Any more than that is high pH. Okay, and then sugar. If you've got more than 2 grams per liter of fermentable sugar, you're at risk. And a lot of people now are even back sweetening wines. Not so much here, but in other areas of the country, like California, they may be back sweetening wines. Okay, so because the consumer likes 5 to 6 grams per liter of sugar. That's a risk, okay? If you don't have very good molecular SO2, which of course ties right in with the pH, right? So if you're at pH 3.5, you need 50 free to have 0.5 molecular. If you're at pH 4.0, you need around 90 free. Don't see many people putting that in their wines, okay? So that's a problem. If you've got potential spoilage microbes, and you know that, that's an issue. And then options at bottling. Many winemakers are constrained by the owner's opinions at bottling as to what they can do to alleviate microbial issues. So some people cannot filter, it's just not an option. Others can't add Velcrin or do things like that, so you just have options. And if you're limited, then you have to recognize the risk at bottling. This is a slide I like to show because it was a Brettanomyces bloom in a bottle. The wine was bottled August 10th, and this is the chemistry. It was, pretty moderate. This wine, by the way, is a Napa cab, so 14.8% ethanol, pH 3.8, glucose, fructose. The wine was pretty dry, right? 0.04 grams per 100 mils, so 0.4 grams per liter. Molecular sulfur, 0.35, not awesome, but not bad. Absolutely no 4EP, 4EG. They ran a Scorpion, 60 cells per mil. I didn't show it, but they also did plating, and nothing grew on the plate. They decided to go with the plate. We'd only been doing Scorpions for a little while then, and they were more comfortable with that data. So then we fast forward to March of 2009, and if you start at the bottom, the Brett had grown up to about 93,000 cells in the bottle. Plating showed about 80,000. They're always a little differential there. But so they had a bloom. 4EP obviously skyrocketed. That's a sensory detectable level. Interesting, glucose, fructose dropped by 0.2 of a gram per liter, or 0.02. So there are people who believe if the wine's dry, it's safe from Brett. No. I think James mentioned that. It's not, okay? So you don't need a lot. And as far as nitrogen, 250 milligrams per liter of yan will support the growth of, a couple 100 million Saccharomyces. So even if you have, say, 2.5 milligrams of yan, that'll support the growth easily of a few 100,000 Brettanomyces. And probably nobody measures yan at bottling, right? Just, it's there, okay? There's always enough there, so not something you can get away from. One of the things that we talked about, or I like to talk about, was screening prior to bottling. Usually what happens is I get this time point. Right before bottling, people run a Scorpion and say, these are my results. What do they mean? Am I at risk? I don't really know at that point. If you got a high level, yeah, you're probably at risk. But this slide, somehow I cannot remove the automatic part on this one slide, okay? The point is, if you have multiple data points, if you're plotting what I call a trend analysis for microbes, you can determine if the population is descending, static, or ascending. You can probably be pretty much certain that if the population is ascending in the barrel, it's going to continue to ascend in the bottle. If it's static, well, then maybe as long as you maintain SO2, it'll stay static. And if it's declining, it's probably declining for a reason, and it's unlikely to increase in the bottle. So once again, trend analysis, a powerful tool. Okay. So what are the tools available at bottling? Physical removal, that's what most people do. Filtration, nominal versus absolute, important to understand that. Just ran a trial last week on some centrifugation, which was pretty interesting because centrifugation was pretty effective at removing Brett, did a good job, not a substitute for filtration. And then additive technology. So your molecular SO2 is what you generally use. You try to get that up there to 0.5 or maybe a little higher at bottling. I don't see a lot of sorbic acid or benzoic acid used on the West Coast. It's pretty big in the Midwest. Lysozyme can be used to drop out gram-positive bacteria. Chitosan products, I think Lalamon's No Brettanside was the first one used in the U.S. It's a very effective tool. Once again, it's not a substitute for sterile filtration, but you can knock down populations by 90% and pull those out. There's other vendors have those products now. There are some for bacteria. I know the product Bactiless, I believe, is both chitosan and glucan, so it pulls out acetobacter. I was so excited when they came out with that product because we finally had a way of pulling acetobacter out of wine. That's pretty cool. But chitosan products, they work, seem to have minimal impact sensorially. And then there's Velcorin, and probably not a lot of people in Oregon use Velcorin. It's really big in California, all right? Velcorin, people have mixed feelings about. Velcorin is called dimethyl dicarbonate, or DMDC. It's extremely toxic. It's deadly to humans. So why would you put that in your wine, okay? Oh, it's deadly to the yeast, too, all right? But let's talk about it. It's not a substitute for filtration, okay? But it's something you add at bottling. It inactivates key enzymes for these critical metabolical processes. So what we see is a slow, or what the company that manufactures Velcorin, this company Lanxess, they call it a lingering cell death. So the cells die slowly, okay? So it's not a magic bullet that blows them away. It doesn't just kill them immediately. I've got a story, I don't have time, but ask me sometime about the people who wanted to stop a white Zinfandel mid-ferment with Velcorin because they wanted to ship it to England and avoid paying full taxes and then re-ferment it when they got to England. Anyways, it's an interesting story, but Velcorin, they tried Velcorin. The thing that most people don't realize about Velcorin, it's only warranted or guaranteed up to 500 cells per mil total, okay? Total. The only way you get to 500 cells per mil total is through very tight filtration, okay? So what I tell people is Velcorin is a very effective insurance policy after sterile or tight filtration, especially if your wine is at high risk, if you have sugar, if you have malic, okay? It's a really good insurance policy. And why can you use it even though it's toxic? It'll kill you because once it gets into the wine, within about 8 hours, it degrades completely into methanol and CO2. So it's widely used in the state that I live in California. It's used around the country. It's used in other industries, perhaps not yet, but if someone decides they want to maybe sweeten their Oregon Pinot a little bit because the consumers prefer that, you might want to look into this as an alternate. One thing to keep in mind is that, as I was telling you, it doesn't kill things right away. This is a graph over time going out from 2 weeks to 6 weeks, and showing after 2 weeks, if we ran scorpions on Velcorin-treated wine, we detected microbes, the Brett in this case, in every one. At 4 weeks, the 3 Cabernets didn't, we no longer detected any live cells there. And then after 6 weeks, it was only in the Pinot, but that Pinot had the lowest ethanol and the lowest molecular sulfur. The Merlot was number 2. The Cabs had higher ethanol, higher molecular sulfur. So it's a synergy effect when you use it. Once again, it's not a magic bullet, but it will probably eventually end up here because it works. So with that, I think we'll go to questions, okay?
And I also wanted to mention, one of the things I run into a lot is the assumption that cross-flow filters are absolute, and I think that's a mistake that Corey, who runs the local service here in Northern Willamette, wants to make sure everyone understands that it is not your assurance, and he's had a lot of clients who've run into issues believing that they've filtered the wine down to a level where there's no microbes. You probably see that a lot. But just making sure that if you want to not go through a membrane, that you're sending it in for testing, because clearly your slide shows even if your sugar levels are very low, you can have issues. So the same person wrote in again for James.
An Irish person, I don't know who that is.
Said, "Show me your Enococcus," which is. We have two. We have 2 questions here. We only have a couple minutes. If someone has an elevated level of acetic acid in wine, what is the best way to remedy with the least amount of invasion?
talking about acetic acid, not bacteria, but acetic acid.
Right.
The only way that I know of that people are doing now is reverse osmosis. Okay, technology's gotten a lot better over the years. It's still a heavy hit on the wine. I don't care what you say. You're separating out, the permeate and running it over a column, which is, removing acetic acid. There's no doubt about it. But what else is it removing? They say it's just acetic acid, but it's a charge-based interaction. And then they're putting that back in the wine. So certainly it will remove acetic acid. Best thing is to not have to do RO. Right?
Could you speak about the effect of botrytis and other microbes on, the botrytis isn't, but on YAN and enzymatic activity?
I think it's, so if botrytis damage on the fruit, I think it's a combination of then having a lot other microbial load coming in, so you're more than likely going to have a reduced YAN, not necessarily from botrytis action, but the fact that it's just now, it's created an environment where everything else now has access to that sugar and it can metabolize a bunch of these compounds and deplete the nitrogen. So it's a combination effect. It's not just botrytis damage. It's everything else that comes after that's going to cause the decrease in YAN that we see.
And we're right at 4:30. Does anyone have a live question? Go ahead.
Well, ideally, stirring the barrel is going to give you the best number. Okay, the question was, do you really need to stir the barrel, or can you just go down to the lees and pull a sample? Yeah. Problem is, if you pull straight from the bottom, you bring up a lot of crud with it, which can then interfere with the ability to do the analysis. So what I recommend is pull 20 mils from the bottom, pull 20 mils from the middle of your barrel and 20 mils off the surface, make what I call a vertical composite, and then send that in. Because if you get too much lees, it interferes with plating. You can't do filtration plating to get any sensitivity because you have a big pile of lees and it follows up the membrane. For in our case, for our scorpions reaction, it interferes with the DNA extraction. And you may see this if you send in a sample with a lot of solids. You'll see that your load detect is no longer less than 10 cells per mil. It might be less than 20 or less than 40, and that's because of solids. So just digging down on the lees, theoretically, yes, that's where the Brett is, but for the analysis, it's more difficult to analyze a sample that has a lot of lees in it.
I think also I'd add that if you're doing the tracking, then if you're sampling from the bottom and then not from the bottom, like the stirred sample is going to give you the best representation for tracking because it's going to be what's in there. If you take from the bottom one time, you might get a lot, and another time you might not. And so I think if you're looking at tracking versus just a 1 point, is it there? Then the stirring gives you that advantage.
Also, if you're doing pre-bottling scorpions, if you're pulling off of the sample valve, which is oftentimes very difficult to actually sanitize, pulling off of the top where you might have your sample tube hit the acetic acid level on top and then drip down into your sample. So what do you recommend for that?
Tank samples, we'll see sometimes where a tank is really clean, very, very low microbe number, and it'll have greater than 10 million Acetobacter. If you're pulling from a sample valve, the urologists have a term called a midstream catch. Okay, so the guys here all figured that, right? All right, and so what you need to do is clean the valve as best you can, because valves are notorious biofilms for Acetobacter. So clean the valve the best you can. Start it flowing out after about a gallon has come out, or whatever you can spare, and then without closing the valve, pull your sample. If you close the valve and then get your sample tube ready and then open it, you're likely to break off some biofilm ineffective. So midstream catch is the best way from a sample valve. Barrel bung, the same thing. The bung hole is a breeding ground for acetic acid bacteria, no matter if you spray ethanol or whatever on there, okay? So just be careful. If you go in with a thief and you're coming out and you touch that edge of the bung, and you put that in, you'll see these really high acetic acid bacteria numbers that aren't real, but they freak you out when you get the results. And then I'll say, why don't we resample and send it in? And usually I don't charge for that, and we get a real number. But sampling is critical.
Yeah, I think we'll end there. I want to thank these two gentlemen. That was really wonderful. Thank you very much.